and then I post a link afterwards for folks that weren't able to join us, or if you guys want to dig back in or something.
You already put the recording on.
I think that's a different one. Yeah. So I'm not sure the Read.ai meeting. That's not me, but I saw that. Is that you, Laura?
I don't think so.
Gotcha.
What I'm recording? No.
Well, that may be a robot that's kind of clinging to my to my laptop. I will if
AI's moving fast.
yes it is.
Beat you to the punch.
Ken, man, I can't. I'm always amazed by your sense of humor, man. So you're always so fast on these things. So yeah, if nobody recognizes the Read.ai meeting, I don't remember signing up for that service. It's not something I'm looking forward to. But I let it in thinking maybe someone else was going to use it to take notes or the like. Does anybody know what the Read.ai meeting is about?
No.
Okay, I'm just going to yeah, I can report it. I can rename it. I'm not sure. It's yeah, a good AI does not allow you to delete it, of course, right?
You
And
can't
so
destroy it.
I'll worry about that later. But I know I did, so thanks, Conor, for mentioning that. So I have now recorded it to a place that I can find the recording, but I may have a duplicate.
If you don't know what that application is, you ought to turn it off because it
Yeah.
might be something that spreads viruses.
Uh-huh. Yeah. I mean, I can even make it a host, David. Yeah, it's really asking for a complete takeover. So yeah, let me.
What does it look like? Might be better looking.
Yeah, so I think it's gone. So,
Okay.
Cool. Um, yeah, so let's. I've got 704. I'm really excited to kind of start a new chapter this evening. So I think we've been exploring, I will maybe say, kind of the engineering side of the glove, and maybe the quantitative side of the glove. Although last time we got into Tango, which is not quite so quantitative, but we did get into some issues about how do we know that something is working? How do we assess Parkinson's? How can we better tune the glove to the individuals that are in the gallery right now? As we age. So that's been the focus, and we've been able to do that by kind of using tools from the outside that kind of measure gait and measure voice, and even measure kind of muscle activity from the surface. But I think to take it to the next level, and also as I begin to onboard more and more students. So I think we maybe you met Alicia last time, and now we have a nursing student from the college that I'm super excited to be joining us. She's really going to help us build the interface for the health assessment. I know what kind of data that I want to gather, but I don't know how to gather it without being intrusive or invasive, or without kind of integrating the larger student group. So Emma, could you just quickly introduce yourself to our group, and anyone else that wants to speak out, or welcome Emma, please, as usual, this is a wide open forum. I do have an agenda, of course, to kind of get through some material, but the main thing is to create a place where we can share. So Emily goes by Emma. So Emma, please introduce yourself.
Hi, everybody. My name is Emma. I am an RN to BSN student at Northern right now. And before this, I was working as a dialysis nurse and had several lovely patients who have Parkinson's. So it's close to my heart. I'm really looking forward to being a part of this project and lending any kind of insight that I can. I'm also very new to research and know nothing about engineering. So I'm just here to help with clinical stuff and expertise. So I'm happy to be here. Thanks for having me.
Thanks, Emma. We had our first exciting kind of session together with kind of brainstorming about how to bring a better health assessment tool where we can kind of gather data while you're at home without even really of course you'll volunteer to offer data, but we can do it in a way we don't have to go to the clinic, and we can do that hopefully in a way that help us tune the parameters of the glove or tune other parts of the whole kind of the four horsemen that Conmore talks about. I mean, so not just the glove, it's the drugs and it's exercise and it's diet. And we need to be super clear about how those things influence symptoms together and but also separately if we're going to kind of tease out what works best for an individual. So thanks, Emma. It's really your energy on Friday was like just contagious. And so even though we had like 20 of the people in the room, it was still a great addition. So that said, we've got a lot of new students coming online, and we have kind of ramped up production. So Aruna, I've got your glove here ready to go out tomorrow or the next day. So you should hear from Laura about trying to get a confirmation of the address either in the States or I guess you're back in India by now.
Thank you so much. Yes, I'm back in India.
So we'll find the best way to get it to you.
Okay, that sounds great.
Yeah, so can we kind of like with that preamble? I mean, I guess it just I've been teaching for 40, 50 years, and I'm a mathematician. And so we tend to, of course, want to motivate why we're going into something as kind of deep and narrow as mathematics. And so I do a lot of preamble to try to motivate people. So maybe we've had like 6 or 8 weeks or 2 months of kind of preamble before going into the more kind of gnarly stuff of neuroanatomy and neurophysiology. But I guess 2 things. For one, I can't resist it any longer, and for number 2, it's drop dead gorgeous. It tells us who we are, what we are made of, and for a teacher, and also for someone with a disease, it's also super positive in the sense that we are not hardwired. We are not hardwired. The glove, I believe, right, is rewiring us, on the fly. We don't quite understand exactly how that's happening, but it's definitely taking advantage of the ability of the brain to be rewired. And so that, without any further discussion, that's kind of what I'm going to call brain porn. I mean, just saying neuroplasticity is like an umbrella term for saying all kinds of things can happen. And we need to be more responsible about what are the networks that we're really trying to change. What are the networks that have been attacked by Parkinson's? And to me, that gives us a great kind of entree into the hard subject of neuroscience. Because when you first open a 1,600-page book on neuroscience, it's just like overwhelming. The brain is just way too complicated. But to go in there to go into our studies with a purpose, to really understand a limited set of networks, and then of course along the way we learn kind of motifs for how the brain represents information. Once we understand those pathways, other pathways become open to us because we have vocabulary and we have an understanding that we can kind of connect the dots. So with that preamble, I'm going to kind of overtake the gallery and share our kind of notes with you guys tonight and step us through the pathways. There's also, I guess there's another issue about where to go first. I think the insides, the up here, the central nervous system is the hard part. We understand a lot about the periphery, largely because we sense it and it's built to be sensed. So we're going to go from the outside in this week, and then next time, 2 weeks from now, we'll go from the inside out. So we'll gather information from the fingertips, get that up into cortex, and then the cortex decides. Basically the cortex has plethora, thousands, maybe millions of motor programs that have been built in since you were small. And it's basically sorting through those programs to decide where it wants to go. Snap the fingers, move, yada, yada, yada. It's making lots of decisions about which motor program to run. And in Parkinson's, there's competition between those motor programs and it's slowing you down. But that's a harder place to kind of figure out what's going up there. So let me, unless there are questions about kind of logistics or where we're going, I'm gonna take over, share the screen. that. So are these the notes that I shared with you guys? Can you see them? And again, just
Yes.
especially for new folks that are joining us. I mean, there is no homework, right? I mean, this is, your receipt of a glove is not contingent on participating in the Zoom session. It's meant to be a forum for us to learn about the brain together and to get feedback about kind of properties of the glove that are helpful or harmful or could be better designed. And so I try to get these notes to you a few days beforehand, but I hope you guys don't see them as homework. And largely today, I just like to kind of share with you some, amazing figures, amazing pictures that help us kind of connect the dots from touch to movement, and from movement planning to better movement or more fluid movement. And so, first of all, is the caveat I think I mentioned before. I can make this a little bit bigger for folks, but maybe not too big. There's lots of hubris surrounding how great mankind is, but to our knowledge, there's no more complex object in the known universe than the brain. 86 billion neurons, each one talking to 10,000 of its neighbors in a way that can change its properties like on the fly. We have fast computers. We have computers with more than 86 billion parts, but they're nowhere near as complex as a single neuron. So again, that's meant to say that if you struggle with this evening's discussion, then like welcome to the club. It's like a really hard problem, but also because of Parkinson's, because of any disease likely. So probably we know more about things when they break, right? It's hard to study how something works until it doesn't work. And then, that kind of the human mind begins to try to take things apart, reverse engineer, poke around. And so oftentimes, and it's a little bit macabre to say that, but disease is a window onto proper function or onto, pre-diseased function. And so it does give us kind of a lens on which to think about the brain. And also helps us focus. We're not going to talk about the whole brain tonight, just a few pathways from the fingertips up. So we're going to talk about touch this week and do movement. Well, we'll see how far we get with touch this week, but I think we can do quite a bit. A lot of subjects have Bibles. The Bible of Neuroscience is Principles of Neuroscience is now in its 6th edition. The last time I was teaching neuroscience, in the 90s and the noughties at Baylor, they were in their 4th edition. Eric Kandel and his band are so this is like 1,600 pages, and it's just an amazingly beautiful text. I would say 90% of it has no serious prerequisites, ever other than a curious mind. They really are, neuroscience is they're bringing in folks, of course, from biochemistry and from biology, but also folks from psychology and from linguistics. So they don't have the luxury, of speaking a very narrow language. The brain does not speak a narrow language. I guess, as an engineer, I can definitely say the brain is electrical. That's a fact, but everything we do in the world gets translated into electricity on the way into the brain. And so if nothing else, if you maybe just kind of click, if you just kind of Google Principles of Neuroscience for 1,600 pages, I think it's less than $130. So it comes out to like, pennies per page. It's an amazing resource. I have I've got a few of their slides, so I'll share with you some of their figures. But we should all maybe have one of these as a paperweight, if nothing else. It's a gigantic tome. We are going to I'm going to jump in. But like I said, often with mathematics, I resist the final dive. And so here's one more bit of procrastination before going into the detailed pathways. Maybe you just want to kind of take a kind of a 3D tour of the brain, not to memorize things, but just as a place where something that we can spin around in our hands, something that we can take apart, and something that we can, kind of get a feel for before kind of peeling away the outer pieces. So I think I've clicked on that kind of 3D brain experience. And I'm not sure if the Zoom can keep up, but can you guys see me spin the brain?
Yep.
Yeah, and I can kind of zoom in and out. Um, and and there's just a little bit of text. It's not too crazy. But if we decided to kind of go in, we're going to be looking at premotor cortex and motor cortex, of course. And lots of stuff tonight is going to even really happening kind of in the midbrain on the way in. An amazing part of where we're going tonight, the major relay station in the brain is the thalamus. And so again, you guys didn't come here to kind of watch Steve click through things. This is a gorgeous, gorgeous brain atlas. And so I can kind of zoom in and out. I can begin to try to rotate so that the thalamus, you've got two of them, one on the right, one on the left. They have a little bridge between them, kind of like your corpus callosum between the two hemispheres. And so these are going to be the relay station. Everything we sense tonight is about touch. Everything we sense passes through the thalamus as a relay on the way up to cortex. And then strangely enough, it also passes back from cortex down to thalamus. So they're like talking to each other while we're not even aware of the fact. It's a gorgeous, gorgeous study. So I take you there. We'll come back to this place. And we'll also begin to kind of play with sliders if you wish. So if those other brain structures were getting in your way, you guys, you can kind of pull them away with that slider and kind of take a more focused look on the thalamus. Of course, the thalamus itself has billions of cells, and so I'm not, but as far as just trying to localize where the action is at, I'll put your brain back together if that felt a little bit, unnerving. Yeah. Thank you, David. There we go. So I so I'm pointing out to you the principles of neuroscience, and I'm pointing out to you this kind of 3D brain map where you can kind of get a big picture. That's to help you from feeling a little bit nauseous as we dive in the next slide. Okay. So the next slide is we begin. I mean, we've already had the big 3D picture, but now we're going to go into the big picture of touch, right? And I can't encourage you enough to kind of go slow and take it all in and to build vocabulary. That's really what we're here for, you guys. And so that we know what we're talking about, but also we need to know what other people are talking about. In particular, there's a lot of snake oil out there, right? And we really want to be clear on what people are claiming. And the more we know about the pathways that are implicated in Parkinson's, then the better prepared we are for discussions with folks that are offering us, a quick fix or something. So I will need to kind of zoom out a little bit, and then I will slowly zoom in. So let me kind of go all the way out, and we'll start at the bottom. So is this big enough for you guys to see? Do you see the fingertip being stroked by a leaf or a feather? You guys can make that out?
Mm-hmm.
Okay. And also, this a lot of figures have too much information. So this one also has kind of parallel pathways that would carry kind of temperature and also pain. Those are of course extremely important pathways. But I just want us to focus in on the red pathway. So the red pathway starts here. We see the finger, okay? We see the fingertip being stroked by a feather, okay? And what I want us to do is count the number of cells, count the number of neurons between the fingertip and the brain. And that gives us something of the map, right? Of how things move. Okay, so there is one neuron. Well, so first of all, there are mechanoreceptors in your fingertips. We'll get to those in the next slide. But I think we all believe there are these kind of receptors in our fingertips that are feeling pressure and temperature and oh, I mean, it's even like undescribable the power of the sense of touch. This is also where a lot of human kindness and love is attached to affective touch. This is a powerful, powerful thing. I will explode the fingertip in the next slide, right? But for now, the fingertip receives information, and we want to see where does it go. So there is this large. There's a cell called a dorsal root ganglion cell. This red one here. That is its red cell body. That where I'm pointing now with my cursor. There's a red cell body with the nucleus of the cell. It's a neuron sitting in your neck, right? It's a neuron sitting in your neck, close to your spinal column, right? And that one neuron sends a fiber from your neck all the way down your arm, past your elbow, past your wrist, right to your fingertip, right? Just one neuron all the way. So its cell body is sitting there in your neck. It sends basically kind of a conduit, a wire, a piece of a dendrite all the way down to your fingertip. But then, of course, it also has a piece of it that kind of gets embedded right there in your spinal column. And then something amazing happens right here at this junction. Okay. So, I mean, I mentioned to you before, right? Each cell talks to like 10,000 other cells. So it's difficult to fathom. So we're not too many here. This cell talks to a receptor on your fingertip, or maybe multiple receptors, of course. But here it even begins to branch. Part of the red business goes up to the brain, and part of the red business stays down here, okay? Stays down here in your spine. And again, we're not gonna go there, but I'm gonna go there because we all kind of know what it's doing there, right? So, are there situations, where you touch, or even worse, you burn? Are there situations where you want to pull your finger back without thinking about it?
Mm.
So this is where a reflex would happen, right? And so information is coming to the spine and being shared with other neurons here that can immediately talk to your muscles and pull back. I don't want to pull back from a feather, but I would like to pull back from this flame, right? So already there's a reflex circuit that's already down there, okay? And I think hopefully we can all appreciate that. We've all, hopefully, I mean, I imagine most of us have pulled our finger away from noxious stimulus. But what's interesting is that reflex circuit seems to be totally fine in Parkinson's. It's not an issue. That circuit, your receptors, these dorsal ganglion cells, they're doing all the right stuff. Okay, the stuff that's in trouble is up in the brain. So you're saying, well, Steve, let's keep on moving. So, any questions about that reflex arc that would kind of start here with that little branch here of the red cell? If that's okay, we're going to follow that red cell up, okay? And he makes his way up the spinal cord into the lower part of the brainstem, into the medulla. And into the medulla, like all other senses, it does an interesting thing, and it switches from the right side of the body over to the left side of the body. And so you guys know that your left brain controls your right part of your body, receives information and controls the right part of your body, and vice versa. Your left brain controls the right side of your body and hears information from the right side of the body, and vice versa. And so there's this. So the cell moves up to the medulla, and there it meets a new cell, right? That does the crossing. So that's where the crossing occurs. The crossing then moves us up through the medulla into the pons, a chunkier part of the brainstem, okay? Into the midbrain, yada, yada, yada. We're just continuing until we get to this amazing thalamus, right? This is the relay station between sensory information and the brain. So as that sensory information makes up to the thalamus just largely, it is just by and large a relay, but kind of a regulated relay because there's this two-way traffic we'll get to eventually. But then from the thalamus, which I showed you in our 3D scan, it then meets another cell. And if we're counting, this is the third cell. We had one cell from fingertip to backbone, another cell from backbone to thalamus, and the third cell, the third neuron, moving us from thalamus to the cortex. Cortex is this outer layer of the brain surface. Cortex is the Latin word for bark. This is the bark of the surface. And this is, another word for this is the gray matter. These are where the cell bodies are located. All the stuff below the gray matter is the white matter. And these are where the axons, where the real wiring is done. But the information is kind of processed up in the dendrites and then whoosh, sent through the white matter to their homies in other parts of the brain. And then there's this, so once you're up in cortex, there is a place, you guys, in each of our brains, there is a place here on the right side and there on the left side for a piece of your body. And so the neurons that feel touch in your leg and foot end up here in cortex. The neurons that feel touch on your trunk are here. Your face is here. Your toes are here. Your genitals are there. Your back and your side. So there's a map of your body laid out on the surface of the brain. I find that an amazing thing to say. But in fact, there are 4 maps of your body. I mean, your body is so important. Your skin is your largest organ. The innervation of skin has to end up in these maps of the territory, and they get laid out in chunks of cortex, right? And so that's the big picture that I wanted to lay out. So let me kind of zoom back out in this. That's the big picture of neuroanatomy from kind of touch sensors, which we haven't really talked about. A touch sensor is innervated by a cell whose cell body, whose nucleus is in our neck. That then kind of crosses over in the midbrain, moves up the midbrain to the thalamus, and makes one final jump into cortex. I find this picture very, very helpful for you guys. If we can maybe look at the side view. I'm also really, really impressed with kind of the medical illustrations that are in. This is from Candel's book. And so the first slice was looking right here in your neck, right at the brainstem. And then the next slice was a couple of these from the medulla. And the next one was in the pons. And the next one was in the thalamus. And then we take a slice of the brain, kind of in this kind of diagonal slice that might take us through somatosensory cortex, where the brain keeps the body. Right here in this beautiful kind of sulcus. And so what we've done is we've looked at cross sections, this cross section, this one, this one, this one, this one, and then finally up into cortex as information from fingertips came all the way up. So there's a lot of vocabulary in the black stuff, right? And again, I don't want to discourage anybody from going there. It's a little TMI for a Monday Zoom meeting. So, and the blue words are mine. So I count 3 neurons, one from the fingertip to the medulla, one from the medulla to the thalamus, and one from thalamus to cortex. These are really, really bulk words, that all of us should have a bead on. Okay. And maybe Steve's gloss is not sufficient if you guys want to go deeper. I mean, again, Cantel's book is the place to go deeper, maybe too deep, but just Wikipedia or the Google on these words, cortex, thalamus, and medulla will maybe give you a better bead on what's going on. But also it's not a bad idea if you're finding, all these extra Latinesque words a little bit kind of, in your face, can you yourself draw a simpler picture? this is something that I would like for my students to be able to do. So if the big picture is cluttered, then remove the clutter and tell me that you really have captured the right stuff. So we're counting 3 neurons, but I haven't really said what a neuron is. It's the building block of the brain. Like I said, we have 86 billion of them. They're in some sense switches. They receive input and then they send output to other cells. There's 86 billion of them. Each one is listening to 10,000 neighbors. Each one probably sends their information to 5,000 downstream neighbors. Again, those are astronomical numbers. And so I did want to just take you briefly into what a neuron, into how a neuron behaves. And I even use this kind of word, kind of white matter and gray matter. And again, please slow me down or ask questions if I'm speaking too quickly or too using words that I've not paused to define properly. So this is a pretty generic picture of like lots of neurons. Well, sorry, of lots of different types of neurons. This happens to be, excuse me, this is just one neuron. To give you some idea of size, like this guy would probably have a cell body about here of about 20 microns in diameter. And maybe from here out to the end of his tree, might be a millimeter. He might be sending his information down a very long axon. And that since that axon might be quite long, he needs to insulate it with myelin. This is fat that really, really speeds up transmission from the spike zone out to its terminals, right? So the idea is that this cell is going to gather input from its neighbors. If it gets enough positive stimulus from its neighbors, it's going to fire whoosh, and transmit that down to its axon terminals where it's talking to its homies. Its buddies are waiting for information down there. And that information could be excitatory or it could be inhibitory. 80% of the neurons turn their neighbors on, 20% turn their neighbors off, right? And again, a lot of diseases are some imbalance of excitation and inhibition. And we'll definitely get to that next week. So the link is there for you guys to play. I'm going to play it quickly. We are seeing information come in through the dendrites and get integrated, and then it decides to fire, okay? So I should like to slow this down, but you guys maybe can slow it down yourself. So information comes in on the dendrites, the cell fires, travels down this white matter axon to its downstream neighbors, right? So this is the building block. This is the neuron. This is the thing you have 86 billion of. And all the action happens here as the cell is listening to its neighbors. It's listening to the neighbors. And of course, just like some of your friends, you have they've got great reliability in giving you, the right information. And other friends, you kind of maybe take their information with a grain of salt. So we have different weights, right, for how we take that information in. Each neuron has different weights for how it's receiving information from its neighbors, based like us, on previous experience. And so it's these weights at these end of these synapses that we're all messing with right now. And as a teacher, that's without those synapses, there would be no teaching, there'd be no learning because we would be hardwired. The physiology of what happens at a synapse, for me, is the most fascinating place in the brain. But we're not going to go there. That's our picture of what's going on in one of these neurons, okay? This root ganglion cell is receiving inputs from lots and lots of receptors and making a decision about whether to send that information to thalamus. Thalamus is a pretty loyal partner in the game. It does not really squelch information. It will relay this, and it knows it came from the finger, so it will send it to the finger part of cortex, okay? It will be modified eventually by knowing what did the cortex decide to do about that, because it's going to send that information back through thalamus on the way to your muscles. Um, so that's the stuff I wanted to talk about in that first slide. And then I have this pregnant question, of course, that should lead us to the next slide. What's really going on at the two ends? So what's really happening at that fingertip, right? And of course, once it gets to the cortex, I mean, that's the interesting part. What's going on up there? Because also this is the part that's being disadvantaged, by the lack of dopamine. So before going to the next slide, are there any? Do you guys have kind of questions or observations about what we've seen so far?
You said the reflex wasn't impacted.
Right. That at least the research I see, Ken, is that the research. I mean, sorry, the reflex machinery seems to be acting, positively, right? There doesn't seem to be any deficit in timing.
Well, there's that movie about Parkinson's patients sort of coming alive, and the actor throws a ball at it and they catch it.
Yeah.
So it
Yeah.
would indicate that Parkinson's, the reflex, still works.
Yeah. 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 I didn't think about that, but yeah, I think that is confirmation of the same. Yeah. So those folks are actually catatonic, right? Yeah. Um, yeah. Yeah. So something else. I mean, that was after L-DOPA, right? That was after.
Right.
Yeah.
He gave him the drug. He
Yeah.
sort of came alive.
Yeah.
Their brain wasn't working, but their hands was.
Yeah. Yeah. Right. 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 Yeah. That's a great point. Yeah. That is kind of, I mean, arguing that the reflexes were there all the time. The doctors just weren't able to get the reflexes used. So um, so I'm also happy to. I mean, again, you don't have to give me lots of feedback, but I also want to be able to take your temperature as we're kind of going into more kind of detailed stuff. If this is too fast, too slow, Goldilocks, you want more information, less information. I will proceed at this pace unless there's, you know, gigantic revolt.
Can I make one more comment?
Yes, please, Ken.
There's another picture in the Peter Tass studies where it shows the brain and the fingers, and the amount of, and it's the first time I ever saw this sketch of the brain, and he shows how much of the cortex the fingers take up, and that's
Yeah.
why he justifies going to the fingers.
Mm-hmm.
And it's that funny drawing with the guy with the huge hands.
Yes.
It sort of shows up in different research texts.
Right. Yeah. So we can, he's called the homunculus, so it's kind of what you would look like, if your body parts were scaled up to equal the amount of real estate they take in brain, right? So
Oh, okay.
you'd have a tiny back. There's very little there. Your legs are not so big. So basically humans would largely be fingertips, genitals, and tongue. That's what we respond to. Yeah. So yeah, it's an enjoyable little monster to see crawling around on, to 3D print one of these. I should do those for some of the students. They're cool to look at. Yeah. So the amount of the amount of real estate in the brain devoted to the fingertips is amazing. And just imagine, right, if there are any violinists in the group, right, or the extra stuff that gets kind of. So also, I mean, cortex is pretty smart. The stuff that you're not using, this myth, you only use 10% of your brain. If you're not using it, another part of your body will grab those neurons and easily kind of, make use of those. So even with a violinist, you can tell right hand from left hand in the brain, just by the amount of real estate devoted to fingertips. So, yeah.
Steve,
Yes.
how fast does a signal propagate down an axon?
Gotcha. Yeah. So that's a beautiful question, kind of the speed of thought. So it goes in tens of meters per second. It's not very fast. I mean, I shouldn't say that. It's relative. Yeah. I mean, we are shorter than 10 meters, so it takes less than a second. But a second of holding your finger over a fire is quite a long time. And compared to sound and to light, these are slow. Because also, if we get into it eventually, conduction is done only through ions, right? So basically, we are still, our brain is still swimming in salt water. So there's this imbalance of sodium and potassium and chloride. The brain is pumping some of those chemicals, sorry, cells pump some of those chemicals out, others in to create a gradient, and then that gives some energy for the cell to fire. And as things move down the axon, the conduction is just ionic. There are no wires, and so it's a really beautiful idea that these fat cells begin to coil around these long axons to speed up propagation. And so I think we'll see some of these in the paper we're gonna look at in the next 15 minutes, Chuck. And they give some ballpark speeds for. In fact, some of the fibers that move from your fingertips to your neck, they're even categorized by how fast they transmit information. So I'll take us back into then the next picture, right? So the question is, what's really going on at the fingertip? Because I mean, again, I don't mean to minimize it. This is a beautiful picture, but this is pretty simple, right? And largely just the fingertip to the backbone, backbone to the medulla, medulla to thalamus, thalamus to cortex, and also there's very little regulation. There's very little kind of decision making along the way. This is a pretty like robust pathway, and as it should be, right? There should not be a whole lot of interruption. Interruption should happen here. What should the brain do with that information? If it's just a light feather, maybe I'm enjoying it. I don't need to move back. But if it's a noxious stimulus, or if it's something I want to lean into, the brain needs to make a decision on those things. But so the pathway I just showed you is pretty unregulated. It largely works. And so I do want to take you into kind of the two ends, right? And so we'll start at the bottom. And in the bottom, this is just your fingertip. And your fingertip has, we're going to shortly see about 4 different types of receptors. And some of those receptors are sensitive to different types of stimulus, to constant pressure, to transient pressure, to fast-moving stimulus or slow-moving stimulus. We have the, where we take these for granted that we can distinguish lots of different things without even seeing them. And so this is a picture that's going to take us from the fingertip. Notice, you guys, it's bypassed your medulla, it's bypassed the backbone. It does take us into kind of 2 places in the thalamus, depending upon the type of receptor that happened in the fingertip. So we'll distinguish between slowly adapting sensors and rapidly adapting sensors. So slowly adapting means that, well, maybe it's easier to say what rapidly adapting is. So rapidly adapting is they learn the stimulus like right away, and then they kind of stop responding. Slowly adapting means they're on board for quite a bit of the stimulus you're giving to your fingertip. Rapidly make a decision quickly and send that information to the thalamus. Slowly are gradually firing, gradually giving you some sensation. So again, these are for this is kind of for rough stimuli, and this is kind of for sharp stimuli, if you wish. Okay. Those are kind of just the 2 basic types of receptors. We'll see in more detail, depending upon on how far we want to go tonight. But already I said what's happening in the fingertip. So there's at least 2 things happening. There are sensors that slowly adapt and sensors that kind of rapidly adapt. I wish I had kind of better metaphors for you guys. So maybe you can think about kind of most of our sensory apparatus in the brain is devoted to vision, like 1/3 of your brain is devoted to vision. And some parts of our visual field, we have really great acuity to things we rapidly adapt to a change in contrast or a change in light, and other things we're more slowly to adapt to. So those sensors are continually kind of integrating information out there as they hand it to us. And so these are two kind of paradigms within the sensorial field. Sensors that are fast and sensors that are slow. The thalamus knows about these two sensors coming from different parts of your fingertip, even though they've got overlapping receptive fields. Oh my God. We'll take a closer look in the next slide. But they get up to thalamus separately. They get up to thalamus separately. And then the thalamus, of course, sends them to cortex. Yeah, and now, so Ken T, now this is maybe gets to one of your points. And so I'm up here now in cortex. Digit 2 is your index finger, digit 3 is your middle finger, digit 4 is your ring finger, and digit 5 is your pinky. These are chunks of real estate in cortex. In fact, they're just in region kind of 3B of cortex. Region 3B has places for all 4 fingers. Oh my God, so does region 1 and so does region 3A. And also region 2. So each of these regions in cortex, this is one of the bulges in cortex, this is one of the valleys in cortex. Bulges are called gyruses and valleys are called sulcuses, right? So that's just good vocabulary to get our hands on. So we've now found our way up from 2 different receptors in the fingertip. Thalamus has been loyal to those as not mixing those 2 stimuli. It's sending them into cortex. And cortex is, this is where all the action is. This is where all the complexity is. This is where all the mystery is. This is where like love and God and hate, all these things exist here as interconnections of these neurons. The cortex has evolved in mammals over time, and at present, I hope we're about to evolve into layer 7, but it takes hundreds of thousands of years for all this to happen. We have 6 layers in cortex, 6 layers, and we happen to know by lots and lots of neuroanatomy and taking brains apart that the slowly adapting cells and rapidly adapting cells even go to different chunks of a cortical column corresponding to each digit, right? So this was someone stroking the 3rd digit, okay? So that 3rd digit is receiving fast-acting stimuli in this part of the column and slow-acting stimuli in that part of the column. These fingers are also represented in other parts of cortex. Right? And so these, and they're right now I've got them kind of going onto 2 different types of cells. The most common cell in all of cortex, most common neuron, is called a pyramidal cell because its cell bodies are shaped like pyramids. Can you guys see that pyramid there? That is the most dominant cell type in cortex. That seems to be the master of them all. It has a very long dendrite. So it receives information from thalamus, kind of down low to the cell body, but it's gathering information from layer 1, layer 2, layer 3, and layer 4, while its cell body sits in layer 5. But the thalamus is also sending information to another subtype of cell in cortex. The second most abundant type of cell is called a stellate cell. And I'm not sure if you can notice, but the stellate cell is actually upside down from the pyramidal cell. It receives thalamic input right at its cell body, and then it sends, this is the axon of the stellate cell, it's sending information up into the pyramidal cell. And so it's this kind of hyper recurrent feedback, all these cells processing information in parallel, which leads to like the main mystery and the complexity of the brain. So I'm going to leave you the fact we know they're kind of 2 cell types. We know different layers in cortex. So can you guys see the map? So I'm going to be back out a little bit. So these are your fingers being represented on a piece of cortex. but cortex itself this bark has many layers, like the bark of a ponderosa. And these layers, largely, the first few layers are kind of incestuous. They're talking to each other. Layer 4 is getting information from thalamus. Layer 5 is sending information to, right? So these are axons now going back to layer 4, going back to the brain structure we're going to talk about next week. So the brain structure most implicated in Parkinson's are a bunch of structures, a bunch of ganglia. It's called the basal ganglia, deep in the brain. But then these are also sending information to the brainstem and even the spinal cord to move your muscles. And then eventually there's this conversation between cortex and thalamus. So thalamus, I said, it's a reliable relay station between your senses and cortex. But then cortex is also kind of double checking these ideas against thalamus. There's this back and forth between the relay and the decider. There's lots of beautiful hypotheses about where the self sits, where consciousness sits. We haven't found the brain region. Many people think it's in these thalamocortical connections. The conversation between cortex and thalamus is what we, perceive as the self or consciousness. There's a beautiful metaphor from one of the early neuroscientists, Charles Sherrington. He called the brain an enchanted loom. And I think he saw as a loom these connections between cortex and thalamus that didn't seem to have anything to do with the senses or with muscles. So this is the brain constructing percepts that we perceive, right? But we don't actually have conscious awareness of that happening. And so just to give you some pointer of kind of the mystery here. So it's complicated. This is not complicated. The fact that your fingers are represented in your brain, I hope they are, right? Because that's how else could that possibly happen? But the fact that there's so much interconnectivity, the fact that a cortical column is receiving and talking to different parts of the brain, and that also in the conversation with the basal ganglia for the Parkinsonian issues, the basal ganglia is also not connected to your fingertips nor to your muscles. So this is an internal dialogue that your cortex is having with the brain structure that interrupts movement. We'll talk about basal ganglia a whole lot next time, as basically the thing that sorts which motor plan we should be executing right now. And without dopamine, that sorting of muscle plans, is compromised. So this is our second major picture, and I'm trying to take bite-sized dives into detail. We went from a fingertip to two different types of receptors on the fingertip. Receptors that are slow to act and receptors that are fast to act. And I haven't really quantified that yet because we're still talking in generalities. So the fingertip having kind of two types of sensors, and I've taken you up into the brain, where there's real estate for each of your digits, and is quite substantial real estate. You're not getting a real picture here. There'll be one coming quite shortly. But then once it gets up there are these cortical columns made up largely of pyramidal cells and stellate cells that kind of process information up and down and across the layers. So that's all by way of kind of introduction to a longer study, which I don't really want to open up to tonight. So I might just stop with one kind of factoid, not unrelated to Chuck's question. So I don't have one on kind of speed right now, but just to get an idea. On the first picture, I showed you this kind of red cell whose cell body was in your neck. It was sending sending a fiber, whatever that might mean, sending an axon, sending a dendrite down to your fingertips. And so there are about 17,000 of these fibers entering the human hand. 17,000 wires are kind of coursing through your wrist and ending up in various places on your hand. We're gonna look at a whole lot of them next time. And then, but not all parts of your hand are equally innervated. Your fingertips have these amazingly dense number of receptors. These cutaneous, so cutaneous, in other words, for skin, afferents, so they're going to receive information and take it to the brain. You got about 240 of these ending per square centimeter at your fingertip, right? And so you can get out a centimeter ruler and take a look at what a square centimeter looks like in your fingertip. It's pretty good size. 242 listeners are down below, sending information to your backbone, from there to the medulla, from there to thalamus, from there to cortex. We're going to take a closer look next time. We're not done with the sense of touch. I want to get into another kind of parallel pathway of touch. So not only does your brain sense fingertip information, but your brain also has receptors in your muscles and in your joints to help us know where we are in the world. I don't have kind of conscious awareness of those, but tango dancers or voice students like Laura, people can learn to know much better, can listen to those kind of deeper sensors, receptors. Those are being processed in parallel with sense. And I'm interested in evidence for how those different pathways are integrated or compromised in Parkinson's. And so I think we got through the first two pictures, but we didn't open up the 5 pictures in that next paper that I was sharing with you. If you can spend some time juggling the 3D brain and finding the thalamus exploding and then kind of putting the brain back together, I think that will give you, kind of more confidence in kind of in where we're going. It may also even help us identify some of the gyri and sulci that are the ridges and valleys of the brain. I wanted to give people a chance to talk about other things that are happening kind of in their world or in our world. So Ken H, thank you for the update. I'm sorry I wasn't able to keep up with all of your activity in trying to get gloves out to a bigger community. But and yeah, I've got my own struggles in trying to make more gloves. We definitely are shipping 10 pair of gloves this week. So that's really what I've been hoping to kind of ramp up to. Largely thanks to young Alicia, who's there in a leadership capacity and helping us make these things. But Ken, if you wanted to share some things about what happened, Ken H, what happened this weekend and for the...
Well, we have the Parkinson Foundation has a walk in many cities across the country, but I walk in the Nashville Move Day, and we had our support group had a booth in Nashville at the walk, and I was demonstrating the glove to people who came up. They put it on, and everybody was interested. We gave handouts that I pulled some information from the website, and then a Dr. Davis from Vanderbilt Medical Center was there. He's quite a lot of these events, and he was aware of Dr. Tass's work, but hadn't heard much lately. But he tried on the glove as I showed you in the picture, and he was very interested. So unfortunately, I think I may have stimulated demand for your glove with him and the people that tried it on.
Cool. Yeah, I mean, we just need. I mean, I would definitely want to involve more clinicians and physicians, right, to try to help us better design clinical trials that can be done, outside the clinic, with you guys just collecting data at home, understanding why this works, how it works better. So thank you, Ken. We'll meet the need. I promise. Yeah. It's a good problem to have.
Definitely.
I have a question about the brain.
Yeah, Laura.
So I noticed that, on a daily basis, I get stiff and all that. But then if I know this sounds weird, but I saw a coyote approaching my cat,
Mhm.
and I just went into total running and
Uh-huh.
grabbed my cat. And I was wondering how was able to have that fluid movement under an emergency, I guess.
Yeah, yeah, yeah. I think that's a great question. That's another kind of window, right, onto how the brain works. So disease is one, but also um, kind of catastrophe or climax is another one, right? And so we talked kind of over and over again that Parkinson's kind of degrades certain pathways, but that the brain is so resilient. There are other ways to kind of to move or to gather information. And so some of these things like just putting kind of a laser beam in front of you or an auditory cue, they can get us to move in a way, that's using it's using a different pathway, right? And so adrenaline is also then bringing on board a whole another set of pathways, right? And so that kind of fight or flight pathway that again, it's not really safe to exercise to be pumping adrenaline into us over and over again. All that's probably been experimented with over time, but I think that's just showing us that there are these parallel networks in the brain, for like I said, the reflexes are there. We talked about with Ken, the muscles are there and in good shape. It's just the brain is actually confused about which motor program to execute, right? And so you're at cross purposes where adrenaline or like DBS is this kind of reset mechanism, which is just giving you the ability that you didn't have without that extra pathway. That's why
Are
that's a very high
you
level explanation. Ken, please.
are you saying that the muscles don't deteriorate as much as the pathways?
Yeah, I guess I'm not. That's a good question, Ken. Yeah, so please, I don't want to speak too authoritatively on this. So the research that I'm seeing is that the muscles, neither the muscles nor the pathways. It's largely happening in something between cortex, thalamus, and basal ganglia. It's
Oh,
the decider. So your cortex has muscle programs for how to move, and how to make these stereotypical movements, right? And those programs are being mixed. So your muscles, which are intact, the pathways that the information to the muscles is also intact. What's missing are the instructions that are on the way.
because I know if I don't exercise for a week, I'm not as good as I did exercise.
Gotcha. Hmm. Hmm. Yeah, yeah.
I
So
just thought that was the muscle, but it's not.
that's a really good point. That's a good point. So yeah. And so I need to think carefully about that. But it's so by my account. So the exercising you do is definitely important for keeping muscle tone, but it could be equally important for actually keeping these motor programs attached to the muscles that you're actually trying to exercise with. Um,
Right.
yeah. It's just that a lot of the literature on kind of the EMG, looking at kind of, muscle contractions, they s- they basically they're trying to find, kind of the key biosignature, and over again they keep on implying, that it's not the muscle, it's not the pathways. Those things, there's almost no distinction physiologically, right, between healthy subjects and Parkinsonian
Hmm.
ones. Yeah.
Interesting.
It's the higher brain function. But that doesn't mean, I'm struggling to make sense of this, but I think I have a hold of it, right? So your exercise, it's kind of back to this kind of proprioceptive stuff. Your exercise is really still reinforcing, the connection between those muscles and motor programs, that are getting shuffled or at cross purposes. And so
Well,
it's
that's
important.
why I've noticed that if I don't exercise but I use the gloves, it's not as bad as if I don't do both.
Mhm, mhm.
So the gloves sort of replicate exercise.
Gotcha. Yeah, that also speaks to one of Conwar's observations, right? Is that he found that the gloves to have greater efficacy, if the wearer was exercising. Um, and that's an hypothesis that I can't tell you how many hours I kind of circle back and forth with
Really?
trying to find ways to better understand that or to tease that out.
Mhm.
Yeah. Yeah.
Nerve stimulation.
Yeah. So you're thinking of I mean, oftentimes for neuropathies, people that have trouble stimulating nerves because the muscle is the nerve is no longer connected to the muscle, the the neurologist can stimulate the nerve, electrically. Um,
Mhm.
yeah.
The other thing is that if in the same system during on periods and off periods, very different things happen.
Yeah.
And during the off periods, a person can move very poorly, but during the on periods, they can move well. So whatever is there is some aspect of it is working.
Yeah. Yeah. Yeah.
There's-
I agree, David. Yeah, that's another thing to say. Yeah, I mean, so the reflexes they must be okay because you're able to the reflexes and muscle control return during the on period. And again, I don't mean to oversimplify it, but during the on period, with dopamine, kind of some part of dopamine back online, you're able to execute individual motor programs without them stepping on each other. Um,
Yeah.
Dystonia disappears too.
say more about that, Ken?
Well, I noticed with myself with carbidopa levodopa, in the morning it'll take care of the dystonia.
Hmm.
So somehow dystonia is things aren't working right. Dopamine gets into the system and sorts it out.
Hmm.
Unless you have too much.
Too much dopamine.
Hmm. Yeah. Yeah. So the balance is another complicated thing. But again, I think that I'm excited about the balance issues just because we're gonna be able to collect so much data if we got the right sensors on your glove and in your phone, and we can kind of give you quicker feedback than wait 6 months to talk to your doctor. I mean, I'm not trying to remove the doctor from the loop. I'm just trying to give you more kind of quicker information based upon your own symptoms.
Mhm.
So
Like getting on a scale.
say again, Ken.
Like getting on a scale.
Yeah.
Yeah, every day versus once every 6 months.
Yes. Yeah. Um, so my plan will be to continue this discussion next time, if that's okay with you guys, and
Good.
we can look into this long paper on touch and proprioception. Um, it's got 5 more figures that largely reinforce things we've already talked about, and yeah, and I'm going to continue to think about some of your questions. And I look forward to future correspondence. Thank you, David Gold, for your email today on kind of on glove redesign questions. We've got Bo also doing a lot of new redesign stuff. I'm so we've kind of ramped up production, but we've also kind of ramped up in people thinking about improvements. So new folks like Mike, I look forward to any feedback you might have, Mike, about things that you're finding uncomfortable or could be better design or a little too flimsy. Do you want
Yeah,
to?
I've got an email I've started working on to give you some things that I'm running into or
Okay,
things that I've thought of.
cool. Thank you.
Mhm.
Mike, if you want to reach out to me, I made some modifications to the glove that was sent to me.
Oh, wow.
Well, I thought the fabric was a little bit too flimsy for me. So, but maybe contact Steve and we'll change email addresses.
Okay.
Yeah.
Yeah.
We'll do. Cool.
Well, thank you, Steve. I appreciate it.
Well,
Yeah, this was interesting.
Yeah.
good. Thanks for taking the journey with me. I'm completely it's just such a marvelous story. What's going on up there, you know, and um, we have a lot to learn, and I think we learn the most if we go in with a purpose, right? And can kind of stay narrow, but still ask pretty great questions, right? So I really appreciate what you guys are helping me with. I'll see
Well,
you.
I will say this, when I was talking to people, I mentioned the rice glove and the northern glove. And I said, what Steve is doing, trying to write an app, and so to getting information from the glove back and so on, I said, it's really leading edge. And people are very interested. Because I explained, instead of just sending out a signal to the fingers, the goal is to gather information and improve things.
Yeah.
While the glove is being worn.
Thanks, Ken. That's the that's the job, right, Emma? That's where we're going.
That
yeah.
is the job, yes.
That is the job. Yes. Yeah.
Has Aaron talked to you yet about it?
No, she hasn't. Did you speak to Erin?
Yes.
Good.
Yeah, we had a nice conversation. She was
Oh,
going to bounce the article off of you before
okay.
she.
No, I haven't heard from her yet. So yeah.
Well, like you, she's probably pretty busy.
Yeah. But I'm glad you got a chance to talk to her. So I'm not sure if she mentioned, but the first press release she wrote about kind of the glove project at the college, she received a national award for best press release in the country for that. So
Really?
yeah. So from little old Northern New Mexico College. Yeah. So it's yeah. So I'm glad that I'm glad, Ken, that you got to speak with her. So that's powerful.
It was a good conversation.
The story continues. I see you guys in 2 weeks.
Sounds good.
Sounds good.
Okay. Thanks.
Okay, thank you.
Thanks so much.